Hello and welcome to my speedrun presentation for Edexcel Combined Science Biology Paper 2. This goes through things very quickly and should not be your main revision but should just be as a top-up just before the exam. So, topic 1, key concepts. Cells are the basic unit of all living organisms. Animal cells and plant cells all contain cytoplasm, nucleus, ribosomes, mitochondria and the cell membrane, and plant cells also contain vacuoles, chloroplast and a cell. The parts of a cell are called organelles, and they include the nucleus, which controls the cell and contains DNA, the cell membrane, which controls what enters and leaves the cell, cytoplasm, which is where chemical reactions happen, ribosomes, which make proteins, mitochondria, which is where aerobic respiration happens to release energy, and chloroplasts, where photosynthesis happens, which contain chlorophyll. Specialized cells are cells that are adapted for a specific role with particular features called adaptations. They include sperm cells, which have a long tail to help you swim, mitochondria to provide energy to swim. They have a haploid nucleus with only half the normal DNA, and an acrosome to digest their way into the egg. The egg cell has a thick membrane around it so that only one sperm cell can enter. It has a haploid nucleus with only half the normal DNA. It has lots of mitochondria to provide energy for growth and lots of nutrients to provide energy for growth. Other specialized cells include ciliated cells, red blood cells, nerve cells, xylem cells, and root hair cells. Bacterial cells have many of the normal features of a cell, including ribosomes, a cell membrane, a cell wall, and cytoplasm. However, they do not have a nucleus. Instead, they have their DNA as chromosomal DNA, this large piece there, and plasmid DNA, a much smaller piece there. Some bacteria also have flagella to help them move. Microscopes. The magnification of a microscope is how many times bigger an object appears than normal. To work out the magnification, you multiply the magnification of the eyepiece lens by the magnification of the objective lens using that equation there. To find the size of something from a micrograph, you measure its actual size in millimeters, then you divide that by the magnification here using this equation. You may need to multiply by 1,000 to convert something into micrometers too. Standard form is a way of representing numbers that are very big or very small. For example, 2.54 times 10 to the power of 6. That 6 tells you how many places you put here after the first number. To have numbers smaller than 1, so they have a negative power of 6, the power this time tells you the decimal point that you start writing your number on, so that's starting on the 6th one because it's times 10 to the minus 6. We did a core practical using microscopes, so we placed a sample on a slide. We stained the slide with iodine to show up the features. We placed a cover slip over the slide to protect it. We placed the slide on the stage, which is just there. We selected the objective lens there, and we used the rough focus, which is here, to get a rough image, and use the fine focus to produce a clearly crisp, sharp focused image. Enzymes are proteins that are found inside every cell, and they speed up chemical reactions. They work on the substrate. The substrate is a chemical with a specific shape that the enzyme works on. Now the substrate fits into part of the enzyme called the active site that has the matching shape for the substrate. The substrate goes into the active site, forming an enzyme substrate complex. The reaction takes place and the substrate turns into the products leave the active site, and the active site is then ready for another substrate to enter. A particular type of enzymes is digestive enzymes. So fat is digested into glycerol and fatty acids by an enzyme called lipase, and that happens in the small intestine. Proteins are digested into amino acids by an enzyme called protease, and that happens in the stomach and the small intestine. And then starch is digested by an enzyme called amylase into simple sugars. The whole point of digestion is to break large food molecules down into ones that are small enough to absorb into the blood. Enzymes can be denatured. Normally the active site of an enzyme has the matching shape for the substrate. However, sometimes the active site changes shape, so it no longer fits the substrate, and this stops the enzyme from working, and this can be caused by high temperatures and very high or very low pH. The core practical on enzymes involved testing the effect of changing the pH on the rate that amylase could digest starch. We did this by monitoring it using iodine solution. Iodine turns black in the presence of starch. So we took a test tube of starch, a test tube of amylase, and the pH buffer, and we warmed them to 40 degrees Celsius in a water bath. We mixed the tubes and started the timer. Every 30 seconds we took a sample out and put it into a little spotting tile well full of iodine, and if it went black we continued repeating the experiment, and if it stopped and didn't change colour, stayed this nice orange brown of iodine, then that meant all the starch had been digested, and we stopped the timer. Then we recorded the time and we repeated this with different pH buffers. To understand how things enter and leave cells, we need to understand the concept of concentration, which is the number of particles in a given volume. If we look at this example here, we can see that this one has a low concentration, this one has a high concentration because there are more particles in that one on the right. Here the concentration of red is lower than blue because there are fewer reds. Here the concentration of red and blue is equal because there's the same number of reds and blues. In this one, the concentration on the right is lowest because although all of these guys contain 7, in the one on the right they're in the smallest volume, so therefore the concentration is higher. Diffusion is how most substances move in and out of cells from high concentration to low concentration down a concentration gradient. You can see that happening in the bottle. The stuff is high concentration in the bottle and it leaves the bottle towards low concentration. This is how oxygen goes from the lungs to the blood and carbon dioxide goes from the blood to the lungs. It's also how carbon dioxide goes from the air into the leaf and how oxygen goes from the leaf back out into the air. Osmosis is how water enters and leaves cells. It is the diffusion of water across a partially permeable membrane. A partially permeable membrane is one like this, which has small holes that are small enough for water to go through, but too big for dissolved substances like sugar to go through. So water diffuses from high concentration to low concentration down a concentration gradient. The concentration of water is higher wherever the concentration of solute is lower. Active transport decides how substances go from low concentration to high concentration up a concentration gradient. For example, how minerals go into roots from the soil or how sucrose enters sieve cells in the phloem. There are proteins in the cell membrane that grab molecules from the low concentration side and move them to the high concentration side using energy as they do so. The osmosis core practical involves taking potato chips of similar sizes, recording their mass and placing them in sugar, sucrose solutions of different concentrations and leaving them for 15 minutes and then reweighing them and calculating the percentage change at the end. What we found was that the ones in the lowest concentrations of sugar increased in size because by osmosis water entered them, and the ones in the lowest concentration of sugar decreased in size because water left them by osmosis. Topic 6, plants, which looks at photosynthesis, the core practical, roots, and transpiration and translocation. Photosynthesis is the way that plants produce glucose using the energy from light. The equation is carbon dioxide and water makes glucose and oxygen, and we also need chlorophyll to trap the energy that's in the light as well. Now, photosynthesis takes place in leaves. Most of it happens in these tall palisade cells with lots of chloroplasts in them. Don't forget though, you've got these air spaces in the spongy layer that allow gas movement, and gas enters and leaves the leaf through the stomata. A limiting factor is whichever factor is present in the least amount, and that limits the rate of photosynthesis. Now, the three of them are carbon dioxide concentration, light intensity, and temperature. The main thing with these, if you look at the graphs on the sloping section, that's when something is a limiting factor. You need to have more of it to increase the rate of photosynthesis. On these flat sections, it's no longer the limiting factor, and something else is holding it back. Also note on temperature, the graph slopes downwards when enzymes start to denature when it gets too hot. In the photosynthesis core practical, we asked how does the light intensity affect the rate of photosynthesis. We took 20 green algae balls and we placed them in some small bottles with some hydrogen carbonate indicator. We wrapped one bottle in foil and left it in the dark, and we placed others at differing distances from a light. We turned the lamp on and waited 30 minutes, and then we measured and recorded the change of the pH of the bottle according to its colour. The one that was bluest had the highest pH. That's because most carbon dioxide had been used up because the most photosynthesis happened, and the one that had the yellowest colour, photosynthesis was slowest because very little carbon dioxide had been used up. And we found that the closer to the lamp, the more the colour changed because the more photosynthesis was taking place. Water and nutrients are absorbed by the plant through the roots. If you look at the surface of a root, it's covered in these little cells called root hair cells that have a high surface area to allow water and nutrients to be absorbed more quickly. From the edge of the root, the water goes into the xylem, which is a tube at the centre of the root that carries the water up to the rest of the plant. The water gets to the xylem either by osmosis, travelling into the root hair cell and then through all the cells, or by diffusion, travelling along the walls of the root hair cells. Don't forget also that minerals can enter roots by active transport as well. Transpiration is how water moves up a plant from the roots, travelling up the xylem, through the leaves. It evaporates out of the leaves and into the air. Transpiration is a one-way movement of water up the plant. Translocation is the movement of sucrose solution from the leaves where it's made through the phloem to the rest of the plant. It is bidirectional, so it can move in any direction to wherever it is needed. Topic 7, coordination, control and homeostasis, where we look at hormones, the menstrual cycle, insulin and diabetes. Hormones are chemical messengers that change the way a part of the body works. Hormones are produced by glands called endocrine glands, and they affect parts of the body called target organs. We've got a whole load of endocrine glands here: the hypothalamus, the thyroid gland, the parathyroids, the adrenal glands, the pancreas, all of them making their own different hormones. Importantly, the testes and the ovaries are also thyroid glands, producing the male and female sex hormones. One important hormone is adrenaline, which triggers the fight or flight response. So this is when we're in danger, we can either fight our way out of danger or run away from danger. Now, this is triggered by adrenaline, produced by the adrenal glands, and adrenaline makes the heart beat faster and stronger to get more blood around the body to the muscles with all the oxygen and glucose, so it can respire more quickly and the muscles can work more strongly. It causes blood vessels going to the muscles to widen to get more blood to them, and blood vessels going to other organs to narrow because the blood is not needed so much by them. It also tells the liver to convert glycogen into glucose to release more fuel for respiration. The menstrual cycle prepares a woman's body for pregnancy. On days 1 to 5, the menstrual cycle menstruation happens, which is where the uterus lining breaks down and leaves the body through the vagina. On days 6 to 13, the uterus lining thickens, ready to receive a potential fertilized egg. In days 1 to 13, an egg is maturing in one of the ovaries. On day 14, ovulation happens when the egg is released. Days 15 to 28, the egg travels along the oviduct, and that's when fertilization is most likely. This is controlled by hormones, estrogen and progesterone. Contraception is aimed at stopping pregnancy, either condoms, which block the sperm from entering the vagina, the diaphragm or cap, which sits at the top of the vagina and stops the sperm from swimming up into the uterus, and the contraceptive pill, which uses hormones to prevent ovulation. The menstrual cycle is controlled by hormones. Estrogen, produced by the ovaries, causes the uterus lining to thicken. FSH, produced by the pituitary, causes a follicle to develop, which matures an egg in the ovaries. LH, produced by the pituitary, triggers ovulation. See the spike in LH there, triggers ovulation. And progesterone, produced by the corpus luteum, maintains the uterus lining and inhibits the release of FSH, so it stops another follicle from developing. Blood glucose concentration is controlled by the hormones insulin and glucagon. After we've eaten, blood glucose concentration increases, so the pancreas releases insulin, and that tells the liver to convert glucose into glycogen, so the blood glucose levels drop. In between meals, blood glucose levels start falling, and so the pancreas produces glucagon, which tells the liver to convert glycogen back into glucose to bring the blood glucose concentrations up again. If you have type 1 diabetes, you do not produce insulin, so you must inject it instead if you still want to control your blood glucose concentration. Type 2 diabetes is acquired during a lifetime by an unhealthy lifestyle and is caused either by your body not producing enough insulin or not responding to insulin. The biggest risk factor for it is obesity, which is a body mass index of over 30. BMI is mass divided by height squared, or having a high waist to hip ratio. The treatments for it include having medication to increase your sensitivity to insulin, eating a less sugary, less fatty diet, and having more exercise. Topic 8, transport and exchange. So surface area to volume ratio, circulatory system, respiration, and the core practical. Whenever substances are exchanged in the body, they travel across the surface, so having a high surface area means substances can be exchanged more quickly. That's why we have capillaries with a very high surface area, and we have lots of alveoli in our lungs, also with a high surface area. We can mathematicalize this using the idea of surface area to volume ratio, which is just surface area divided by volume. Higher surface area to volume ratio means substances are exchanged more quickly. Your circulatory system is your body's transport system, carrying oxygen and nutrients to the cells and carbon dioxide and waste away from the cells. It is made of your arteries that carry blood away from the heart under high pressure with their thick muscle walls, mostly oxygenated blood. We've got veins carrying blood towards the heart under low pressure with thin muscle walls, and that's deoxygenated. And they've also got these valves in them as well, so blood only flows one way. And we've got capillaries with their very high surface area to allow substance exchange to happen very quickly. Now, in your blood, you've got red blood cells, erythrocytes, with a biconcave disc shape with a high surface area, no nucleus to allow them to carry more hemoglobin. We've got white blood cells, which fight disease, so lymphocytes make antibodies, and we've got phagocytes, which engulf the pathogens. And then we've got platelets, fragments of cells, which clot the blood. And we've got plasma, which is the straw-coloured liquid here that carries the blood cells and transports dissolved substances. The heart is a double pump for pumping blood. The right side of the heart pumps the blood to the lungs and back to gain oxygen and lose carbon dioxide, and the left side of the heart pumps blood to the rest of the body to lose oxygen and gain carbon dioxide. The top chambers of the heart are called the atria, and the bottom two chambers are called the ventricles. In between the atrium and the ventricle, you've got these flaps called valves, which make sure the blood only flows in one direction. The amount of blood the heart pumps every minute is called the cardiac output, and that is the stroke volume, the amount of blood pumped by each heartbeat, times by the heart rate. That's the number of heartbeats every minute. Respiration is the release of energy from glucose. Aerobic respiration takes place in the mitochondria and involves glucose and oxygen becoming carbon dioxide and water. Now, during exercise, as our energy demand increases, we need to do more respiration, so the heart and breathing rate increase to get more oxygen to the cells to allow that to happen. Anaerobic respiration happens during times of intense exercise when we need an energy boost on top of aerobic respiration, and that involves glucose turning into lactic acid. The lactic acid is poisonous, so we cannot do this for a long time. In the respiration core practical, we investigated how temperature affects the rate of respiration of small organisms. We placed some soda lime in a test tube. Soda lime absorbs carbon dioxide. We put 10 maggots in the test tube. We placed a bung with a capillary tube in the test tube and left the whole thing in a water bath for 5 minutes to get up to temperature. Then we dabbed a little red ink spot in the end of the capillary tube, and we measured how far the ink spot travelled in 5 minutes. As the maggots respired, they used up oxygen, replaced it with carbon dioxide, but the carbon dioxide gets absorbed by soda lime, so the whole volume decreases, and that's where the ink spot moves along. And we found that the higher the temperature was, the further the ink spot moved, because the faster the respiration was taking place. Topic 9, in which we look at ecosystems, biotic and abiotic factors, measuring populations, and nutrient cycles. An ecosystem is the organisms and the environment in which they live. It's made up of a community, which is all the organisms living in an area, the populations, which are the members of a single species in an ecosystem, the habitats, which are different areas of the ecosystem, and all these food webs, which show the feeding relationships in an area. Now, if we look at the food web, we have the bottom level is called the producers, then the next level up is called the primary consumers, then the secondary consumers, and so on. Note the direction of the arrow shows where the energy goes from and to. Now, to estimate the number of organisms in a habitat, we do random sampling. So we take some quadrats, we drop them at random, and then we count the number of organisms in the quadrat. Do that a few times to take an average, and then the total population is the organisms in the quadrat times by the total area divided by the area of a single quadrat. We did a core practical called doing a belt transect to investigate how the number of daisies varied as we moved away from a tree. So we took a quadrat, we placed it at the base of a tree, we counted the number of daisies, then we moved it a metre away, counted the number of daisies, moved it another metre away, counted the number of daisies, and so on. And we did that until we were 20 metres away from the tree, then repeated that with several more trees, and we took averages, and we found that the further away from a tree, the more daisies there were, because the more light was available, the more water, the more nutrients, because the tree wasn't using them. Biotic and abiotic factors affect what can live where. Abiotic factors are non-living influences on what lives where. For example, the temperature, the general climate, amounts of rainfall, the soil type, the presence of pollution. Biotic factors are living things that affect what can live where. So the presence of plants for food, the presence of prey to eat, the presence of predators that might eat you, the presence of plants for shelter, the amount of disease, and so on. Parasitism is a feeding relationship in which one organism feeds off another without benefiting it. The way, for example, leeches suck blood, the way mistletoe puts its roots into the trees of another, the branch of another tree, and the way tapeworms feed off our guts, and so on. Mutualism involves two organisms working together for mutual benefit. For example, cleaner fish and sharks. The little fish here don't get eaten by the shark because they do the shark a service. The way that coral and algae live together as well is another example of mutualism. Now, biodiversity in humans. Biodiversity is the number of different species living in an area, and the higher biodiversity, the better. Now, humans have had a big negative impact on biodiversity, for example, by introducing species into an area that have no competitors. So, for example, cane toads in Australia having a big negative impact on biodiversity because nothing eats them. Fish farming causes lots of pollution, and again, that reduces biodiversity. Eutrophication, which is the effect of fertilizer running off farmers' fields into watercourses. Sometimes, though, we can improve biodiversity as well by conservation, for example, captive breeding and reforestation. These are things that we can do to increase biodiversity. The water cycle is how water moves around the environment. It starts with precipitation, so rainfall, snowfall, etc. That rainfall is collected into rivers and lakes and things like that. And from the rivers and lakes, it can evaporate into the air, and it eventually condenses to form clouds. Those clouds then lead to precipitation, and the whole thing goes around the cycle again. Now, water is treated to make water potable. Potable water is drinking water. The main parts of it are filtration to remove dirt and things like that from the water, and chemical treatment to kill pathogens that might be in the water. The carbon cycle describes how carbon moves around different parts of the environment. So there are big stores of carbon. Carbon dioxide in the air the carbon stored in living things, carbon stored in the soil. Now, carbon dioxide gets from the air into living things by photosynthesis. It's moved from living thing to living thing by feeding relationships. Living things also respire and produce carbon dioxide, putting carbon back into the air. Sometimes as their bodies die, the carbon goes back into the soil, and some of that can even end up in the form of fossil fuels buried deep underground. And sometimes we burn that and turn the carbon dioxide back into the air as well. The nitrogen cycle describes how nitrogen moves around different places in the environment. So it starts off in the air and gets into the soil through the work of nitrogen-fixing bacteria, which turn it into something called nitrates. Those nitrates can be absorbed by the roots of plants and used to make proteins, and then the plants get eaten by animals, the animals get eaten by other animals, and so the nitrogen moves between animals and plants quite easily. When those animals and plants die, the nitrogen they contain goes back into the soil, and that can either be turned back into nitrates, or denitrifying bacteria can turn it back into nitrogen gas, and it goes back into the air again. Thank you for listening. The end.